back and talking more about lipid biosynthesis. In this lecture, we're going to be looking at the continuation of our use of fatty acids after the actual initial synthesis of our base fatty acid, palmitate, how we can elongate that, how we can desaturate it, for example, to use it in other forms. And also we'll be talking about then how lipids can be modified into some bigger, more complex structures like phospholipids, which are used in cell membranes, and also the synthesis of other types of lipids that are not utilizing fatty acids, in particular steroids. This is a lipid class that doesn't use fatty acids at all in its structure. Just a reminder of kind of where these processes are taking place and the different types of processes associated with fatty acids. Remember, we spoke about the initial fatty acid synthesis that occurs in animals. And this is going to make our base fatty acid, which is a 16 carbon long fatty acid called palmitate. Now, palmitate can then be modified into a longer fatty acid, or it can have double bonds added in, for example, and this all occurs in the endoplasmic reticulum. We'll be talking about that today. I want to point out that another thing that occurs in the cytosol is the base formation of steroids. Our other lipid class that doesn't utilize fatty acids can also be synthesized in the cytosol. The final portions and modifications of steroid formation also occurs in the endoplasmic reticulum. And you'll note if we want to do something more complex, like form a phospholipid and make these more complex structures using the fatty acids, this also occurs in the endoplasmic reticulum. So again, if we want to take palmitate and lengthen it, which is a very common process, this can occur in the endoplasmic reticulum. There are also some mechanisms for lengthening in the mitochondria as well. Now, lengthening can only occur by 2 carbons, can only occur in even numbers, and this is why, for example, animals only utilize and break down fatty acids of even number. We can only break down into units that make acetyl-CoA, which happen to be 2 carbons long, and we can only build up with units that are 2 carbon long because we're utilizing acetyl-CoA and malonyl-CoA. So one of the most common products from lengthening is just the addition of 2 more carbons onto our base palmitate. Palmitate can be lengthened into stearate, which is 18 carbons long and is the most common product. Now again, what if we want to desaturate a fatty acid? What if we want to add in double bonds? This might be useful, for example, in relation to fluidity. Remember that saturated fatty acids are more solid. They stack better, and these are not as fluid. If you wanted to have fatty acids that allow for more fluidity, for example, in a cell membrane, then you would maybe want to have some more double bonds in there to kind of kink the structure and allow for more fluidity when these start to pack together. To add in a double bond, we're going to have to use an enzyme called fatty acyl-CoA desaturase. The desaturase is the main name here I want to point out. This is noting that it is desaturating the lipid. You can also see that there are cytochrome B5 that are utilized in it. So this desaturation is occurring through the redox reaction of an iron in a cytochrome B5. Ultimately, though, this reaction is reliant on the input of an NADPH to then add in that double bond. So not only is NADPH useful for the formation and synthesis of fatty acids, it also is going to be used in desaturation reactions if we want to add double bonds into our now formed fatty acids. Some other important lipids that can be synthesized are echinosoids. These are hormones, and they include prostaglandins, leukotrienes, and thromboxans. These are lipids that are used as signaling molecules. And ultimately, they are formed from our base fatty acids. So fatty acid synthesis is still important for the formation of these echinosoid hormones. And these can then be created into a base hormone lipid called arachidonate. This is going to be, again, a base lipid that can then be used to make all kinds of prostaglandins, leukotrienes, and thromboxans. These can be incorporated into membranes, or again, they can be used for signaling. For example, prostaglandins are extremely important for signaling in inflammation. In fact, one of the main mechanisms of our anti-inflammatory NSAID drugs is to target the synthesis of prostaglandins to help reduce inflammation. So an interesting note is that these NSAIDs are going to target a enzyme that is responsible for the catalyzation and formation of prostaglandins. There is COX-1, which catalyzes the synthesis of prostaglandins that actually help regulate our gastric secretions, the mucus in our intestines and stomach. There's another enzyme called COX-2, which catalyzes the synthesis of other prostaglandins that are important for mediating pain, inflammation, and fever. Now, again, a lot of NSAIDs inhibit COX-2 particularly, and they're going to try to target COX-2 to stop the formation of prostaglandins related to inflammation and pain. Now, unfortunately, some NSAIDs are not as specific to COX-2 and can also target COX-1. COX-1, because it is important for our stomach and intestines and the protection of them through mucus, if it is inhibited, this can be eroded at. That's why a lot of NSAIDs, if they take too many of them or you take them too often, you can start to develop things, problems with your stomach. You can have some scarring or ulceration that occurs because you're accidentally targeting COX-1 as well and removing that gastric mucus that is needed for protection. This is one role of prostaglandins, their importance, and how they are related kind of in the world of medicine. Here's kind of a schematic of the inhibition of that COX. Cyclooxygenase is the name of the enzyme. it's abbreviated as COX. So cyclooxygenase activity, again, is inhibited by certain NSAIDs. One of the well-known ones is aspirin. And you can see that it does this as an irreversible inhibitor. Remember from Biochem 1, irreversible inhibitors bind in a fashion in which they do not let go of that enzyme once bound. And essentially what they do is block the site for the true isozymes that should be binding onto COX. This is for aspirin. Ibuprofen and naproxen, other NSAIDs, are competitive inhibitors. So they resemble the substrate. They resemble the actual molecule that normally binds to COX, and they compete for that site for binding. Okay, so let's talk about some of the other more complex structures that could be made once we have synthesized fatty acids. One of these is triacylglycerol. This is useful in the storage of fats, and also phospholipids, which are very commonly used to help make cell membranes. Both of these type of complex structures rely on a molecule called glycerol. We've seen this when we talked about the breakdown of lipids. When we break down triacylglycerols or when we break down phospholipids, glycerol is one of these products that happens from the breakdown. Remember, this is usually the backbone portion that is holding these together, and glycerol is a 3-carbon molecule. Each carbon having an OH group on it. This is a base glycerol. And you can see that through these alcohol groups, these OH groups, we can bind on fatty acids. This is also the same point in which the fatty acids are broken off of the glycerol when we're doing oxidation. So if glycerol is needed to make triacylglycerols or phospholipids, it has to be synthesized. And just like the reverse, when we're breaking down fats and we get extra glycerol, it is usually turned into glycerol 3-phosphate. In reverse, when glycerol is needed, glycerol 3-phosphate can be used to make glycerol to help make tags and to help make phospholipids. Glycerol 3-phosphate, again, is an isomer. It's in constant balance with our other molecule, dihydroxyacetone phosphate. These both come from glycolysis. Remember, they're a step of glycolysis. We make glycerol 3-phosphate and dihydroxyacetone phosphate. They are isomers of each other in balance. You can convert one into the other through a isomerase. But glycerol 3-phosphate is what is used to make glycerol via glycerol kinase. This reaction, you can see, is going to use ATP to make glycerol 3-phosphate if we're making glycerol. And so in the reverse, if we're going to take glycerol 3-phosphate and make glycerol, we actually generate some ATP. So when glycerol is converted into glycerol 3-phosphate, we use ATP, and the reverse, when we are taking glycerol 3-phosphate and making glycerol, it generates ATP. Now, what if we want to synthesize a phospholipid? Phospholipids, again, are used widely in cell membranes all through the body. They're very important structures. and similarly need the backbone of glycerol and then the fatty acids as well. So one base type of phospholipid is called phosphatidic acid. This is probably the most simple of the phospholipids that can be made. It's a precursor to our triacylglycerides. Essentially, glycerol 3-phosphate is taken and it's not converted into glycerol, just glycerol 3-phosphate on its own, then has 2 additional fatty acids attached to it. So here's the glycerol backbone in pink. We're going to keep the phosphate on because phospholipids have phosphate groups on them. So no need to break it off into glycerol. And the other 2 spots, right here and right here, are going to have fatty acids added onto them. You can see they're shown in blue. They're abbreviated. Remember, these are long chains that are sticking off of that. So this is the easiest phospholipid you can make. You're taking a glycerol 3-phosphate and you're adding on 2 fatty acids using ATP per fatty acid that you add on. So we have to use ATP for this stuff. Phosphatidic acid can be then used to go down other pathways to create other more complex molecules. This is a way that you can also make just base types of triacylglycerols by just removing that phosphate. So this is another way you can make the di or triacylglycerides. Instead of making glycerol first and then adding on fatty acids, we can make phosphatidic acid and then remove that phosphate group. If you remove this phosphate group on its own into an OH, that makes a diacylglycerol, DAG. And then if another fatty acid is added on, we make a triacylglycerol. So this is another way we can take our base molecules and build up diacylglycerols or triacylglycerols. Now, if you wanted to make any other type of phospholipid, we've talked about phospholipids. They have a base of this phosphatidic acid, but attached to the phosphate can be other novel head groups. Depending on what that head group is, you'll have other types of phospholipids. So instead of phosphatidic acid, PA, you could have phosphatidylinositol. Phosphatidylcholine, for example, is another one. These are all just based on that head group portion. Changing that around will give you a different type of phospholipid. So these can be changed out just on that phosphate, resulting in different types of phospholipids. So phospholipids can be synthesized in this way, where you start with a phosphatidic acid, as I just said, and you then change out the head group. Or you can also go through another route where you take diacylglycerol. If you happen to have some diacylglycerol around, this might be from a breakdown. There's also a path that lets you convert it into a phospholipid. So another path allows you to take diacylglycerols and attach a phosphate on, condensing this phosphate with a head group in one go. So you do a double condensation, you add a phosphate group on, and you add a head group on to that phosphate, ultimately making a phospholipid. This is another pathway. If you happen to have diacylglycerol around and you need to make phospholipids, you need to synthesize them, this is one route that can be taken. So an important factor of phospholipid synthesis is this head group, changing these head groups out and adding on different head groups. To add on a phospholipid head group, it requires the activation of cytidine diphosphate, CDP. So CDP is particularly important for phospholipid synthesis. Now again, you can start making a phospholipid by starting with a diacylglycerol that is then activated by cytidine diphosphate, CDP, and attaching a head group to it. So you can see here is our base diacylglycerol. It has the phosphate and cytidine attached to it. So this is cytidine diphosphate is attached. This is helping to activate this position, allowing for the addition of a head group to come in, ultimately breaking off this portion here, leaving cytidine monophosphate, CMP, and resulting in our now phospholipid with a phosphate and a head group attached. Another way to do this, again, is to take a cytidine diphosphate that already has a head group on it, and essentially doing a similar process. You're going to activate the CDP first and then add it onto the diacylglycerol. So either the diacylglycerol is activated with a CDP and then a head group binds, or the CDP is activated with the head group and then binds onto the diacylglycerol. This is how phospholipids such as phosphatidylcholine and phosphatidylethanolamine are made, through this process using the activation of CDP with a diacylglycerol. So this is an important role of a nucleotide outside of DNA, outside of RNA, of cytidine diphosphate in lipid generation and phospholipid specifically generation. Okay, so now let's talk about our other types of lipid structures that don't rely on fatty acids. These are related to, but distinct from things like tags, phospholipids, sphingolipids, and plasminogens. All of those I just noted use fatty acids as their base. Cholesterols, steroids, these all are going to instead use a building block called isoprene. So this is a 5-carbon building block. You can see it here. That is what helps build the actual backbone of cholesterols and steroids. Cholesterol biosynthesis goes through a long process. It's a multi-step process, starting actually with an acetate. An acetate. This is then formed into a mevalonate, this long molecule here, which ultimately is condensed through the addition of isoprene. So we start with some building blocks and then add in our isoprene. And isoprene is used and activated through phosphate to then start building this backbone portion. These polymerize through a multi-step process. This is shown here in a very abbreviated form in which things are cyclizing and adding together, ultimately going from a molecule called squalene. Squalene is kind of an intermediary molecule that leads to the ultimate formation of cholesterol, which you can see here. And I'm just showing you this again because the last slide really abbreviated those steps. In no way do you have to memorize these steps, but it's nice to see all of the processes that are happening, going just from squalene, where we have already put together these isoprenes. Notice that squalene doesn't actually have rings yet. It's still a chain, it's just kind of bent and kinked, but it is not rings yet. Now we're going to use NADPH again. This is an important molecule even for the synthesis of cholesterol. And this can then help to start cyclizing the molecule. Now, squalene can help to form other base molecules, not just cholesterol, but other steroid-based molecules that can be used in other types of organisms. So in plants, for example, stigmasterol is a common product. Ergosterol is a product in fungi. But in animals, we're ultimately going to go through this process of making cholesterol. Now, after cholesterol is formed, cholesterol can then be further edited and changed to make other similar molecules of a backbone that has the cholesterol backbone. It's synthesized in the liver and then ultimately exported, usually as things like bile acids or cholesterol esters. So bile is going to be stored in the gallbladder. This is helpful for breaking down fats that we ingest. So we're using a fat acid, essentially a ionic form of a fat, to then break down other fats that we're ingesting if we just ate a fatty meal, for example. Other tissues convert cholesterol into things like steroid hormones and other hormones that can be used for signaling in the body. So these are going to be things like your sex hormones. Here's the breakdown of cholesterol into things like bile acids. Taraccolic acid is one very important bile acid that is used in the intestines, for example. Cholesterol synthesis is also regulated, the transport of it and synthesis of it. In particular, you can see here is our multi-step reaction, starting with acetyl-CoA. Here's our cholesterol. This can then, of course, be turned into things like cholesterol esters, which we just saw, and other forms of cholesterol. There's LDL cholesterol, for example, which is our extracellular form that is able to be transported through the blood. Now, molecules such as insulin and glucagon are regulators of this pathway of forming cholesterol. In the case of insulin, you can see this activates the pathways to help form cholesterol and cholesterol esters. Glucagon is going to be a signal to turn off cholesterol synthesis. Now, these act particularly on this enzyme called HMG-CoA. HMG-CoA is one of the most important regulatory points related to cholesterol synthesis and formation. So there are many ways that HMG-CoA is regulated. Some of them I just spoke about, for example, through activation through insulin or deactivation through glucagon. There's also transcriptional regulations of HMG-CoA, and also another gene, ACAT, as well, which helps to increase the esterification and storage of cholesterol, is another area that is regulated. And lastly, I wanted to note another application of medicine and drugs in relation now to cholesterols and steroids. So a very, very key drug, statins, actually resemble mevalonate, one of those molecules in the stages of forming cholesterol. And mevalonate is the product of HMG-CoA reductase. So it is involved in the enzyme HMG-CoA reductase. Again, the main enzyme that regulates this whole process. Statin resembles mevalonate, and it actually competitively inhibits HMG-CoA reductase by binding. It will compete against normal mevalonate and bind into HMG-CoA, reducing it and ultimately lowering cholesterol synthesis. This is how statin is used as a cholesterol drug. For people with high cholesterol, statin is often prescribed to help lower their cholesterol by acting as this competitive inhibitor. Statin was actually found first, it's called lovastatin, the molecule, in fungi. It was isolated there. This was a natural product that just happened to bind to HMG-CoA, and so it was utilized as this competitive inhibitor. Because it does this, it helps, of course, with lowering cholesterol in patients that need it to help improve circulation. It stabilizes plaques that people might have, again, by removing cholesterol from them and reducing vascular inflammation. So it has been a very important drug, a very high money-making drug as well, because it is helpful for high cholesterol and those who have heart problems related to their cholesterol as well. Now, statin is going to generally disrupt cholesterol synthesis, so it does have other downstream effects. It ultimately is going to affect, for example, cell membrane formation, because cholesterol is an important component of cell membranes. Cholesterol is still a needed molecule. This is why we don't want to inhibit in a way that completely stops cholesterol formation, but only is kind of lowering cholesterol formation. So there is a balance here. Okay, well, that is it for our lipid biosynthesis. We've talked again about general ways that we can form some more macromolecules of lipids like phospholipids, triacylglycerols, and also cholesterol. And in our first and previous chapter, we talked about the base formation of our fatty acid building blocks through our fatty acid synthesis enzyme in the cytosol of cells.